And now I'm going to move into introducing our second and final plenary session of the day. This topic is called the Therapeutic Toolbox. So I'm happy to introduce Dr. Carsten Bönnemann. He is the head of the Neuromuscular and Neurogenic Disorders of Childhood section, NINDS, from the National Institutes of Health. Also, Dr. Alan Beggs, director of the Manton Center for Orphan Diseases Research at Boston Children's Hospital, and Dr. Gustavo Dziewczapolski, scientific director of Cure CMD. And I'm excited to learn about genetic therapies that are being worked on for each of our subtypes. So let's dive in. Thank you.
Thanks for the introduction. Let me see, am I muted? No, you can hear me, right? Okay, so what we're going to do today is a little bit of an experiment. So we see how well we do with that, but we're going to try to really introduce some of that miraculous technology that you've all been reading about and see how that could apply to the diverse disorders that we cover here at Cure CMD. And we're going to do that by the technology and not by the disease, and see how that applies to the different technologies. So let's see how that goes. And feel free to chime in with questions. So Dr. Megan Foley will be monitoring your questions, and Dr. Dziewczapolski and Dr. Beggs will be commenting, running commentary as we go along, and hopefully we cover some of those questions as well in our commentary. So important, we're going to talk about gene and transcript-directed therapeutics here in the session. So not about medical care, not about small molecules, and things of that nature will be discussed in the research or clinical care sessions. This is really the kind of gene-directed therapeutics that we're going to discuss today. We're going to attempt to provide an overview, probably not going to be comprehensive nor precise enough, but we're going to make a best face effort to cover this, and then we can always ask questions. So we hope that you will have at the end some idea of where things are applicable, and how we can maximize efforts in therapeutics development in ultra-rare diseases such as ours to really get the most mileage out of these exciting approaches that are happening at the moment in the field. So here's a paradigm and the promise of gene-directed approaches to disorders in general. The idea is our disorders are genetically caused. They're caused by gene mutation. And the closer we get to the origin of the disease, the higher we expect the impact to be. And so this is illustrated here in a very simplistic diagram, but it's still helpful of the flow of genetic information. Our chromosomes are written in DNA, and that's where the genes are in DNA. Then a process happens that's called transcription, which is basically reading the genes from the DNA, making them into a messenger, which is called RNA. We call that the transcript. And the RNA then is the template to make protein, and the protein then makes a person. Now, in a genetic disease, we have a problem right here in the DNA. So something is messed up, something is different in the way the genes are written, and that gets carried forward, that gets transcribed into the transcript, and then leads to a faulty or even absent protein, and that leads to the disease. And the idea of what we're discussing today is the closer we get to the source of this, to the mutation, the higher the impact. Now, that's not to say that working down here at the impact of the disease on the patient isn't important. As a matter of fact, nothing is as influential, as consequential for our patients than working on the impact of the disease. Medical management still outperforms anything that we have tried up here in the genetic medicine. So it's really important to not forget that. But for the session today, we're trying to move to the source here. So what is a precondition for that? So the precondition is that we really have to know the disease down to the gene and mutation level. So in order to be precise about genetic treatments, we have to be precise about the diagnosis, even down to the mutation level. So I always say, precision diagnostics, that's a precondition for any precision therapeutics. And therefore, it is really, really important that we work diligently and very hard that every patient in our community has a full and complete genetic answer to the disease they're dealing with, because we want to have that ready for the promise of genetically directed therapies. And don't panic, this is the toolbox here. I'm just putting that up to show you that there's lots of things are happening. Up here is the things that you can achieve. These are the different tools that are at our disposition. And we're going to talk about some of these along the line here in this talk. But the idea is it's a toolbox. So you have a screwdriver, for instance, that fits certain types of screws just perfectly well. And then it doesn't matter which engine uses the screw. If you have the right screwdriver for that screw, you can fix a number of different engines with that. And that's the principle of a toolbox. You have a tool that has a certain genetic target, and as long as the genetic target is shared by different diseases, you can cross-apply it and therefore be much more efficient in developing therapies for even ultra-rare diseases. That's the idea behind the toolbox approach. And the one in dark blue are the ones that are probably closest to clinical translation. We talk about all of them, AAV-mediated gene therapy, ASO, and siRNAs, and other things that you would see. So let's talk about AAV at the beginning. So Dr. Foley made a great point that AAV is not adenovirus. It's actually called adeno-associated virus. And you can see that here, this big guy here is an adenovirus. So that's what is used in the vaccine development. The little guy there, that is an adeno-associated virus. And it's called adeno-associated virus because it hitchhikes on the hike with the adenovirus. It needs the adenovirus to provide proteins for its own replication. So they only exist in a collaboration. And fortunately, adeno-associated viruses are largely non-pathogenic to humans. They just kind of come along for the ride. They live to replicate and spread around, but they don't cause disease. So that's a really good feature of them. And so that's why they are so useful for human gene therapy. So they have their own genes here. And as you will see, we're going to replace the genes the AAV uses by the genes we want to deliver as a therapeutic and just take advantage of its ability to infect cells, deliver something, and then don't allow it to replicate. So it's a kind of one delivery tool that we have. So it is naturally occurring. There are many serotypes that are naturally occurring that are in clinical use right now, but we can also engineer it for our purposes in the future. The big disadvantage of this tool is that what you can package into it is size limited. So when you replace the genes of the AAV by the gene you want to deliver, I call that the transgene here, you're limited by the size the AAV will allow you to put in there to be still competently packaged. And this is how you do this. So now you have replaced the viral genes by your therapeutic gene, but then you have to supply the genes that the virus needs to package itself completely in separate portions, its own genes, and the helper genes from the adenovirus are now supplied in the cell system, and that allows the AAV to be packaged as a virus, but it has no viral DNA in it. It has the transgene in it, and that's your therapeutic product. So you have a natural viral envelope, but an artificial viral genome which contains your therapeutic gene, and so it can infect, but it cannot replicate. So it infects once and then goes away. However, for the immune system that looks at it from the outside, it looks just like a virus. And so the immune system will make antibodies against it. But as Dr. Foley has pointed out, this is not adenovirus. So the antibodies that you develop in a COVID vaccination that will not interfere with AAV. However, there's a certain degree of natural infection in the population. So some of us run around with antibodies against various serotypes of AAV, and therefore this gene therapy approach cannot be used because the immune system is just going to take it out and not allow it to infect cells. We come to that in the discussion. So AAV is a versatile tool. Because of this flexibility of the system, you can package loads of things in it. So you can replace a gene that's missing, but you also can load it up with other components of our toolbox. And you'll see that later, how that can be useful to deliver other things, including gene editing tools, just taking advantage of its packaging capability and infection capability. Now we're just talking about gene replacement. So a gene is missing, and we're using the AAV to replace it with an artificial gene that makes a protein that's lacking. And it's done in a simple construct. You have a driver of the gene that is determining where the gene is expressed. And here you have an optimized version of the natural gene that is made in a way that it does the best job it can possibly do in making the protein that's missing. So this is best for situations where protein is actually missing and where the gene is small enough to be packaged into this vector delivery system. And that, of course, causes opportunities, but also limitations of this approach. So let's talk about gene replacement. Here's my little diagram again. So the concept here is you have your AAV. It has packaged your optimized replacement gene in it. You make it infect the cells where it's needed. So for instance, the muscle cells. It delivers its extra DNA to the cell, but most of it will not integrate into the chromosomes. Most of it lives in the cell, but it has its own little genetic element. We call that an episome. Very little integrates into the genome. Most of it stays outside, but it makes a transcript, and that transcript makes a protein. And that protein is now able to replace the protein that's missing, and we hope for a therapeutic effect coming from that. So that's the idea of a gene replacement therapy. But because it's not in the chromosomes, it will also not inherit it from cell to cell. So when the cell now starts dividing, where you have delivered the DNA, the daughter cells will get very little of the goodies. And so you can dilute it out if the cells start dividing and get bigger. And that's a problem, of course, in muscle disease. Here is a prime example where this could really work very well, which are the alpha-dystroglycanopathies, one of the subtypes of CMD that we deal with. As you know, we have loads of different genes here, but they're all small. They're all small, and they're all diminished function or loss of function. They're really almost all fit into the envelope and could be delivered, and then very little goes a long way for them, probably. And the dream here would be to have one delivery tool, one vector, one way to deliver this, but then just flip out the gene that's needed, and you would have what we call a platform approach. One gene therapy approach that ideally would deliver many different genes that cause alpha-dystroglycanopathy. Now, this is music of the future, but that's the only way we can get at the ultra-rare disorders. And we're working with UNCAD's National Center for the Advancement of Translational Science to develop such a thing. We call that PAVE-GT, platform vector gene therapy. I think it's a great opportunity to address these ultra-rare diseases that are very kind of related, where you just have to deliver a different cargo for the same type of disease. So here are the genes that would benefit from that. The alpha-dystroglycan, almost all genes in alpha-dystroglycan would benefit from there. And there's some very exciting data that was just presented last week that shows this is quite feasible, and very little goes a long, long way in gene correction for the alpha-dystroglycanopathies. The recessive mutations in collagen 6A1 and A2 would also be addressable, because those two genes are still small enough to fit into AAV. However, A3 is too big, and selenon. So unfortunately, COL6A3, LAMA2, COL12, also Titin, I didn't put that on the list here, are too big to be replaced by straight gene therapy. And then sometimes you have additional complications here in LMNA, for instance. You have to also knock out the dominant version of the gene, and we'll talk about that later. So that's a short introduction in AAV replacement therapy, and now I'd like to ask our panel to comment a little bit on what they see as the promises, the limitations, the considerations for the different diseases they're interested in.
So thank you, Carsten. That was a wonderful overview. And I would say I agree completely. There's tremendous promise here. As with any new technology, there are clearly challenges and limitations. The limitations you mentioned regarding gene size and the fact that it needs to be a disease caused by the absence of something are well understood. Other things that have come up are potential toxicity related to the infusion of very high doses of this. So while we understand that the AAV virus is non-pathogenic, doesn't cause disease under normal circumstances, in some instances we've seen that delivery of very high doses in a therapeutic environment can actually lead to a bad response. And so this is certainly concerning, but the positive thing is that we're starting to develop and understand ways of tweaking the virus so that you can provide it in lower doses and achieve greater delivery of your therapeutic gene while putting in lower doses of virus. In this case, we can minimize those risks. In addition, understanding those risks, there are various drugs that can be given and procedures taken at the time of treatment to minimize bad reactions. Other areas to think about are how do we regulate the gene that's included? So in some cases, the early studies that are being done use a promoter or a gene switch to turn on this gene that is expressed widely throughout the body. In other cases, it's expressed only in some tissues, for example, the heart and the skeletal muscle. And as we develop more approaches to this, we understand better ways of turning this on or off in certain places. That's going to be very important in the future. For some neuromuscular diseases, we probably want that therapeutic gene to be expressed only in skeletal muscle. In others, for example, selenon is expressed throughout the body at low levels. And we're still trying to understand, during development, how it's turned on and off. So we'll be able to control this much better in the future. We also need to consider the possibility that expressing too much of that therapeutic protein might not be good in certain circumstances. And so as we understand that better, again, we can control it. And then finally, you mentioned the immune responses. And so there are certainly immune responses to the vector, to the virus itself, and those can be modulated. There are companies developing approaches to kind of hide the virus proteins when it's infused into the body, or to combine them with things that turn off the immune response, that block T cell responses of your immune system. And so that holds promise in the future. And the ability to retreat. As you mentioned, these viruses are found normally in the population. And so many of us, particularly as we get older, experience an infection with a certain AAV serotype. You mentioned serotypes. They're basically just different versions of the virus that the immune system recognizes as being slightly different. And so the serotypes usually have a number. So AAV 1, 2, 3, 4, 5. In a neuromuscular disease, AAV 8 or AAV 9 are used. So many of us already have antibodies to AAV 8 or AAV 9, but there in the future may be ways to blunt the response to that or hide the response. And if you've received a treatment and the treatment has worn off over the course of a span of years, it may be possible to retreat either by cleaning the immune system through something called plasmapheresis or hiding these epitopes. At any rate, we have a lot more to talk about, so I'll leave it there.
That's a great point, because in some of our children, of course, these being congenital disorders, we want to treat when they're infants, ideally, and very small. And so they will, of course, make more cells as they grow, and then our virus will be diluted out. And we have to have a way of retreating, because gene therapy is a very effective way of immunizing against that particular serotype AAV. But there are several ways and technologies now underway to see whether we can sneak that by and make redosing possible, including switching serotypes.
Absolutely.
So I have,
I can just add a few numbers that in the last decade, 40 clinical trials involving the use of AAVs were in development, and about more than 600 children in viral gene therapies have been treated. So we are gaining a lot of experience in those areas that Alan and Carsten mentioned.
Yeah, and Regan already mentioned nanoparticles, and just to kind of give some hope also to the large gene cohort here, so the large collagen, titin, there are nanoparticles under development that are capable of packaging messenger RNA, so transcripts of quite a larger size than the limited packaging capability of AAV. And here you could use a nanoparticle, deliver the messenger RNA directly. This would lead to its own translation make a better protein. Disadvantage, of course, here is this is transcript, not a gene, meaning you would have to redose it to provide transcript, as it's not being read of a gene, but it is provided by the nanoparticle, so that it would have to be redosed. But in any case, the dosing capability, the packaging capability is much better, and nanoparticles are now, of course, in millions of people as part of the Pfizer and Moderna viruses, vaccinations, which are using nanoparticles. So the safety looks really good for these. So let's talk about another idea here for the large genes, which is functional gene replacement. So the idea here is if my gene doesn't fulfill the size requirement for AAV, maybe there's something else there that can stand in for it, even in the body's own genome. And we know from Duchenne muscular dystrophy, for instance, that we have such a backup gene, which is utrophin. We know from spinal muscular atrophy that we have a backup gene, which is called SMN2. And we know now that we have a similar backup gene also for laminin alpha-2, which is, of course, the root cause for our LAMA2 cohort. And so, as you know, the LAMA2 gene, when it's missing, does not allow to be made into merosin. And then we have a different laminin hanging out in the extracellular matrix, which is not capable of making the proper connection to the muscle. It's just sitting there, not doing the functional job. And so there are a couple of ways to try to get around this, to reestablish the linkage here with replacement genes. And one of them is to use a shorter version of the body's own backup genes to reestablish the linkage here, using a mini-agrin and a mini-nitrogen here, which is able to interact with this puny non-functional laminin 411 and reestablish a linkage. You hear much more about that in the research section we have coming up. But here the good news is both of these small body engineered, if you will, replacement proteins fit into AAV, and that would now be amendable to gene therapy. And the functional gene replacement here, so where we have a gene that makes up for the function, we talked about LAMA2, but you can also consider that for alpha-dystroglycanopathies, where you can think about a gene that would just add glycoepitopes to dystroglycan, no matter what's missing in the first place, and reestablish a linkage that way. Those are the two opportunities for that. So maybe we can briefly comment on the considerations and limitations of that.
Sure. Actually, first, let me address a question from Dr. Shamindra Konersman, who was asking about dystroglycanopathies and pointing out that many of these affect both the brain as well as muscle. And so how would we approach that? So my quick answer to that is you can control it two ways. You can modify the virus itself so that it's more likely to infect cells in the brain as well as the muscle. And you have to consider how you cross the blood-brain barrier. And right now for spinal muscular atrophy, for example, the injections are actually being done into that compartment of the body. But there is development of viruses that are more likely to go there. And so you might have either one virus that can go to both places, or possibly even in the future, two different vectors. And then, of course, yes, as is being asked, would you what would you do for the promoter, the signal that turns the gene on? And you can imagine promoters that would turn on in both brain and skeletal muscle, which is what you might want for some of those.
Yeah, most definitely. And you can even think about alpha-dystroglycan being a protein that's pretty widespreadly expressed in the body. We call that ubiquitous expression. You can even think about a more ubiquitously acting promoter to allow for that to happen. And in the central nervous system phenotype in alpha-dystroglycanopathies, I don't think is beyond the reach of gene therapy. Of course, if there's a severe brain malformation, that we can't turn back. But there is also probably dysfunction of the way the neurons talk to each other. The synaptic connectivity is dependent also on alpha-dystroglycan. It's not inconceivable to think about reestablishing some of that connectivity by supplying alpha-dystroglycan to synapses, making that better. So I think that jury is out, but I think there's hope for that too.
Yeah. And then regarding functional gene replacement, clearly for each of these approaches, it's critical to know what the defective gene is, and that's how we base our development of therapy. And in the previous example, we were actually replacing a missing gene. In this case, we're turning on or providing something else that compensates. And so in order to do that, it's really critical to have a good basic science understanding of what we call the pathophysiology. What are the mechanisms of disease? What is the pathway or the network of proteins that interact with each other, so that you can make an educated guess as to what you might need to tweak in order to compensate or make up for the primary mutation. So when you talk about studies in zebrafish or mice or cells that sound rather esoteric, they're really critical to developing these kinds of therapies.
That's exactly right, for the proof of principle of it, and I'll get right into that here. So in the previous functional gene replacement, we used kind of functional genes that we engineered to replace what's missing. And now here's an approach to use the body's own genes by just regulating their expression to get the same thing done, which is to replace the alpha-2 chain here with the alpha-1 chain. And then you get a version of laminin that's called laminin 111. And very careful genetic experiments in mice have shown that the alpha-1 chain, if it is re-expressed, if it's pumped up where it's normally not expressed, is able to re-establish that link as well. So here we have a complete body rescue gene that we just have to churn up to do the linkage here. And you can do that by a protein replacement that's being tried right now. It's in preclinical development to replace the entire laminin 111 as a protein. But today we talk about gene-directed therapies. So for that, we will have to go and see what we can do on the genetic level. So in my little diagram, I have a little cartoon for everything here. So now we are delivering a machinery that is able to regulate genes, and it makes use of a technology that we describe later, which is the CRISPR technology, CRISPR-Cas technology, which allows us to basically, like a search function in a word processor, to find a particular thing in the genome, log onto that and deliver something, either a gene scissor or in this approach here, a gene regulator. So now we use that to go to the nucleus, find another gene and turn it up. So in this case, here is our LAMA2 gene that's defective, but now this machinery goes and finds the LAMA1 gene and turns it up to see whether we can replace functionally the LAMA2 gene. And that would lead to a LAMA1 protein down here, which should be able to replace the LAMA2. And so this, again, will be discussed in greater detail in the research section, but Dr. Kimala Devi in Ronnie Cohen's lab has shown that very nicely. So here you can see how similar the two genes are. And here she is directing her gene regulatory machinery directly to the LAMA1 gene and turns it up to replace the LAMA2 gene. So more to come. So there are also other possibilities to gene regulate, many of which are not yet explored. And Alan mentioned how important it is to really provide proof of principle in animal models to really see how impactful that is. So here is an example of CHOP and ERO1 regulation in selenon. I don't know, Alan, whether you want to comment on that.
Actually, we may hear about that a little later this afternoon in the selenon session, so I'll leave it for
you. Okay, so here you're basically using our gene-directed machinery to regulate genes that make life easier if one gene isn't fully functioning. So now I want to come to a different level of interfering with the flow of genetic information or correcting it, which is not at the gene level itself, but which is at the transcript level. And you've already heard a little bit about that when we talked about the lipid nanoparticles, because here we delivered a transcript to the cell. But there's also a number of highly advanced technologies, some of even FDA approved in other in other indications such as Duchenne muscular dystrophy, where you can direct your therapeutic to the transcript and modulate what the transcript is doing and even correct abnormalities in the transcript. And that we call that transcript-directed approaches, which are really the the 40, the strengths of oligonucleotide therapeutics, and I will show you that in a second. Why is that still necessary? Well, so some of genetic conditions cannot be addressed with simple gene replacement. We talked about that. The gene may be too big, or the genetic mechanism may be different. And there are certain types of mutation that really lend themselves, they really cry out for transcript-directed therapeutic approaches. I'll show you an example of that, where that is possible. And then also small molecules, ASO, are much more cost-effective to produce and make at high quantity. So that allows us to even think about making a precision therapeutic for a single patient. It's not inconceivable to identify a mutation that could be addressed with an ASO and make that just for that patient. So that's the ultimate precision or boutique approach to therapeutics, but it is less of a futuristic approach than it was just a few years ago. And so we should talk about that, and also talk about how important it is to find the exact mutation in every single patient to see whether it could be amendable to this. So back to my cartoon here. So now we have a custom-made little oligo that we now give into the body, not with an AAV, just as the oligo, like a small medicine that's usually given by injection into the body. And that goes to the cell, but it now goes not to the gene, it goes to the transcript. And sometimes will correct the transcript, lead to a better protein and a therapeutic effect. Let me give you an example. The best example is splice modulation. And splice modulation happens when the gene is read, made into the transcript, and the transcript has to be cut to size to be made into protein. Now we call that process splicing, and a lot can go wrong in the splicing process. Here's a great example from the recent work that we did in collaboration with many groups in the world, where we identified a mutation that interfered with the normal splicing process by introducing a thing to the transcript that doesn't belong there, a pseudoexon. That all of a sudden gets put into the transcript and is highly toxic and messes everything up. But we can use small ASOs, small oligos, to hide the splice event from the splicing machinery, to suppress the splicing, to not allow it to be spliced into the transcript. It gets skipped out. It's pseudoexon skipping, and we generate a normal transcript again. So that is therapeutic splice modulation that is precision made for this particular mutation. And it's not crazy. It actually works. And here's another more complicated example from the alpha-dystroglycanopathy world. In Japan, where Fukuyama congenital muscular dystrophy was first discovered by Dr. Fukuyama, it turns out all cases of Fukuyama congenital muscular dystrophy are based on the same mutation, where a jumping genetic element happened to jump right into the fukutin gene. And what it does there, it also messes up splicing. A message can no longer make sense to the cell and can't make normal fukutin. And what the researchers, and so it creates this dysfunctional misspliced version. And what the researchers in Japan have done is they used oligonucleotides to really mask the abnormal splicing to the splicing machinery. It hides it from the splicing machinery, and now the splicing machinery will do the right splice and get a naturally normal transcript reestablished. So another precision splice modulation approach, which hopefully will go towards clinic as well, at least for the patients in Japan. And there are many of them because it's a common form of congenital muscular dystrophy in Japan. So that's a CMD mutation-specific RNA-directed splice therapy. And really, I could put every gene in our box on this list because every gene, in particular the large ones, in particular things like titin, LAMA2, COL12, the large COL6 genes can have abnormal splice mutations that can be potentially addressed with these splice modulation therapies. So that really means we really need to drill down to the actual diagnosis, even if it's not evident from the primary testing. And Helen, can you comment on that, how that's best done?
Yeah, I mean, of course, the big difference between this and what we've talked about previously is the fact that this is not a general approach for a particular gene, but it really is specific for a particular mutation. And even the same type of mutation in two different genes require different different reagents, different oligonucleotides to target the specific sequence. And so this is where molecular diagnosis is really critical. In the past, to find these gene mutations might take years. And some of folks on the call who we've worked with waited 5 years, 10 years before the mutation that caused their condition was discovered. Nowadays, with newer so-called next generation sequencing approaches, it's possible to sequence the majority of our important DNA sequences responsible for disease very quickly. And in many cases, roughly a third to maybe, in good instances, a half of cases, we can actually identify mutation. Knowing that mutation can allow us to do that. And it's really, for those individuals who have a mutation amenable to this, as Carsten said, the development can be remarkably fast. So, for example, my colleague Tim Yu here at Boston Children's Hospital identified a girl with a unique mutation in a gene that caused a condition called Batten disease, a severe neurodegenerative condition, and was able to, within 1 year, identify the mutation, design oligonucleotides specifically for her sequence, only working for her mutation, nobody else on the planet that we know of, and to get approval from the FDA. And ultimately, sadly, she passed away, but her disease course was significantly slowed down for a while. And the approach that was developed there is one that can be applied across the board. And of course, when you have recurring mutations like the COL6 mutation that Carsten's group identified, or the Fukuyama congenital muscular disease mutation, then these approaches can be applied to larger numbers of patients. So personalized medicine is often the moniker that people give to this approach. It's incredibly powerful and is rapid. Our ability to roll it out in a time frame that really benefits people is rapidly increasing.
Yes, and the FDA seems to be cognizant of that as well, and they're really working with us to really develop pathways towards even single patient treatments. But as Ellen was mentioning, some mutations are actually recurrent, such as the intra-mutation in COL6, where it makes more sense. Gustavo, any comments?
Again, maybe mentioning about platforms, as you mentioned, for alpha-dystroleukinopathies and working in the vector, the viral vectors, is also a possibility to think about platforms to address a specific mechanism of mutation and how to repair that. And so, again, put it in parallel instead of in line to work on those.
Yeah, and there are crazy applications of that. For instance, in Titan, you could say, well, maybe we can engineer by splice modulation a Titan isoform that's still functional, but ignores certain mutations that occur to it, similar as to the dystrophin approach. So then you would have a cocktail of splice modulation that would force Titan to be a different isoform that excludes a mutation, for instance. So that would be another cool approach. Regan, any comments from the audience or questions?
I was going to say, if anyone who is watching right now wants to ask their questions as we're going through the therapeutic toolbox, please feel free to post your questions and we'll feed them to the panel to answer as we go along.
Let me just make one last comment about this. As Carsten rightly pointed out, this approach theoretically might apply to any gene that causes disease in humans. But he's mentioned the larger genes like Titan, also, for example, the ryanodine receptor or nebulin mutations, which cause Nemeline myopathy. Those are the ones more likely to have the type of mutation that's amenable to this. And so the smaller genes like selenon, SEPN1, some of the others may be better candidates for replacement therapy because most of those mutations won't be the type that we can approach by this method. They're not going to be altering splicing.
Most definitely, yes. So another forte of transcript-directed therapies is to inactivate mutations. So now we're in the land of the dominant negative mutation. Those are important in the collagen, they're important in element A, where you have one mutation that creates a gene product that interferes with the normal gene product, and you have to get rid of it. So collagen 6 disorders are a prime example of that. If you have a mutation at the beginning of collagen assembly in one of the three subchains of it, and it gets carried forward in the assembly, it is able to poison most of the collagen 6 product. And if you were able to take that out right at the beginning and not allow it to be assembled, then you have 50% of a normal collagen 6, but in the case of collagen 6, that doesn't cause disease. So you have to be precise, of course, in activating just the mutant allele. But that really is in the works at the moment and is a possibility. So prime candidates for this approach are collagen 6 or 3 genes. There are certain mutations in collagen 12 where that approach just works just as well. And then we have two kind of more complicated candidates, which are LMNA. And I mentioned here FHL1 as well, because I know we have a family on board here as well, where you have these dominant mutations as well that you need to inactivate, but half of LMNA or half of FHL1 actually, or in FHL1 cases, not having no FHL1 is not enough. So you need to replace and repair at the same time. And we get to that in a second. So that's a little less straightforward, but still possible. And the way you do these mutation-specific knockdowns is by using tools that recognize single changes in a transcript, such as sRNA or certain oligonucleotide approaches as well. Still the land of small ASO, small oligonucleotides therapeutics.
So once again here, this is dependent on understanding the precise mutation. And in the simplest approach, you design that siRNA or that oligonucleotide to bind and inactivate, for example, only the abnormal or mutant version of the messenger RNA. However, that's obviously, as before, very patient-specific and unique only for that mutation. Turns out there's a lot of normal variation in our genes. And so in some cases, if it's not possible to target the actual mutation itself for technical reasons, or if you'd like to design a drug that might work for multiple different mutations, we can actually design those drugs to target normal variation in the messenger RNA. And when you sequence a patient, we all have 2 copies of each gene. If you find that they've got one version inherited from mom and one version from dad, and there's normal differences, normal variation between them, you could target, for example, only dad's version because you know the mutation is on the version that was inherited from dad. So by targeting that normal variation that you know is linked together with the mutation causing disease, you have a reagent actually that can work for more than one patient and be more general. So this is just one of the tricks that people are developing to try and expand the applicability of this.
And Regan is conveying a question from the audience here about an umbrella approval of ASO-siRNA technology. And that's in the works on the level of the actual chemistry of it. So this would mean you have a class master file, if you will, for a certain chemistry of ASO that allows you to say, well, we know the chemistry, we know the toxicology of the chemistry. Now we only need to make sure that particular sequence is safe on the genetic level and doesn't cause abnormal splicing elsewhere. And that saves you lots of animal toxicology that is very, very expensive. Hopefully we'll get there. We have 5 minutes left, I see here, and let's see where we go next. We'll go next to CRISPR-Cas. I think everyone is probably wanting to hear a little bit about CRISPR-Cas. So that's one of the gene scissor system that you've seen in the press, but it's much more complicated than that. So CRISPR-Cas is a very ingenious way of finding certain spaces in the genome and acting on them in various ways. It is developed from bacterial immune systems, actually. But again, it works like a search function on a word processor, where you have, this is your search term here, this little nucleotide, and goes into the genome and finds a certain spot that you're interested in, and then brings this machinery here, the Cas9, that's the actual gene scissor, to that particular spot in the genome, and it allows the Cas9 to make a cut there. And so now you have to see what you make with the cut. You can leave the cut as it is and just kind of interrupt the gene there, which is a possibility. For instance, if you want to knock out a gene, you just kind of go there and you just cut, and then it's knocked out. So that's a possibility for the dominant disorders. But, and that's where it gets really exciting, you have also an opportunity to repair, to put in a replacement patch that has now the wild type and no longer the mutation sequence in it. So extremely powerful concept. where you would deliver the goods again with an AAV. These are large things, so you probably need 1 or 2 AAVs to do that. You go to the nucleus, the CRISPR finds the spot in the genome and it does a repair there ideally. So it's on the genome level. And again, the group of Ronnie Cohen with Dr. Kemala Devi, who's going to report on that, has done a proof of principle in a llama 2 mouse by cutting and re-annealing a piece in the llama 2, a gene to get rid of a particular mutation. But that's purely mouse specific. So there's no human mutation that could be treated with this. This is just proof of concept of that. We've done the same for our pseudoexon. You can take your gene scissors and just cut out the pseudoexon out of the intron there. Just get rid of it. Cut it away. Don't repair. Just cut it out. And you no longer have any pseudoexons, and you have repaired the sequence there. So that's powerful. And then Gustavo supplied this example here for LMNA. You go to LMNA, you inactivate the LMNA gene as a mutation, so it can no longer be made. So it's now kaput, it's broken, but at the same time you repair it and then you knock down and repair. So that approach, genome editing, is essentially available for all genes. But of course, there's lots of considerations and limitations. And I will have our panel weigh in and have the last word here. And since we're out of time, we're going to leave base editing, which is kind of a refinement of this technology, for another time. And we'll finish up here with CRISPR-Cas. And I'll let our panel have the last word here.
I don't know if we have time to just address. There are 3 very good questions in the chat. So I'm going to just see if we can address maybe at least 2 of them. One is, how does one get a single mutation therapy?
Regulatory-wise? Yeah. So is that the question?
I believe so. It just was quote unquote, how does one get a single mutation therapy?
Approved, yeah. So it's still a long way and a very complicated way with lots of regulatory approvals that are necessary. At the moment, this is probably easier for the ASOs than for the gene therapy because you need less toxicology probably on that level. And once you have shown proof of principle in cellular system, not even in an animal model, in cellular system, you have evidence that this is not genotoxic, not systemically toxic. You can actually make oligo in a way that's called GLP. So it has to be really high quality and FDA accepted, and then go back to the FDA for a single patient approval and IND. So in Tim Yu's case, that took close to a year, I think, from the discovery to the patient, and set them back about $1 million. And so that process can be made cheaper and shortened, but it's possible. For gene therapy, it's also not impossible, but it gets much more expensive and much more complicated to do. So the idea is behind PAVE-GT and other efforts is to smoothen that regulatory process, to make take advantage of master filings and cross-referencing to make that process cheaper and faster.
So I think we're out of time, but I'll finish with one long sentence about the promise of CRISPR approaches, which is tremendous. There's a lot of excitement about this. There are 2 main challenges. One is the efficiency. So how well does it work? How many cells in the body can be corrected? And the other is off-target risks. In other words, is this therapy going to alter some other gene in the body and cause something bad like cancer? But there is tremendous work in this area. And so it's something that we all are excited about and look forward to advances in the future. And with that, I guess we should probably wrap it up. But Carsten, that was spectacular. Thank you.
Yes, thank you. And then we can move the discussion in each breakout room.
Yes, exactly. I was just going to say, please, in the breakout rooms, you find the experts there, and you can continue the discussion. And we could probably spend hours here, but Rachel is not going to let that happen. So I better stop here. And thank you all for, and Sarah also not. Okay, thank you.
Thank you.